Okay, so kinetics and reduction oxidation, we are almost finished. We're just going to really go quickly over oxidation and reduction, probably something that we've all been learning since high school. But as I'm sure you've all noticed, when you get to organic chemistry and biochemistry, it becomes a little bit more complicated and sometimes a little bit more difficult to identify. So we should also talk really quickly about equilibrium. Delta G, that little mark above these deltas just means under standard conditions. So when we take standard conditions, we can calculate standard conditions, and it's very helpful for us to actually calculate the delta G in practice that's actually happening in the reaction. So delta G equals delta H minus T delta S obviously represents the equation for Gibbs free energy at standard conditions. So if we add a little prime there, which is usually what happens when we're talking about biology or medicine, so that prime is the free energy at physiological pH, because with standard conditions, all solutes are usually at 1 molar. But if we're talking about pH, hydrogen ions are not at 1 molar in the human body, definitely not. So we adjust to physiological pH and we just use that prime to mark, just to separate it from the regular standard condition delta G. But it's basically the same thing. You can use them you can't use them interchangeably, but these formulas, these next 3 formulas that we're talking about, anywhere you see that delta G standard, you can use that physiological pH delta G as well, and the formula will work. So we can relate the free energy of a system to its equilibrium constant. So that delta G, if you can't get it from this formula, delta H minus T delta S, you probably won't be able to, because a lot of times they won't give you the actual standard number for enthalpy and entropy. You would use this equation, which is negative RT ln K EQ. So you should probably memorize all of the formulas on this page. It would definitely be helpful. You will probably have to use that formula. So our negative RT ln of K equilibrium. So R is the universal gas constant, which will probably be given to you on test day. There are a couple of them depending on what units you use. And then you just multiply that by the ambient temperature in Kelvin, obviously. And then you take the natural log of the equilibrium constant, which we denote as KEQ. So KEQ is just the concentrations, the product of the concentration of the products over the product of the concentration of the reactants. So KEQ, this second one, to be a little bit more correct, there should be subscripts on each of these concentrations. Outside of the bracket, there should be a little EQ, just like there's on the K. But it's the same formula. You would plug in the concentrations of the products and then the concentrations of the reactants to get the equilibrium. So it's basically just the ratio of products to reactants once the system reaches equilibrium. So when we know that, we have all this information, we can actually calculate delta G of a reaction that we're running. So delta G is going to equal that standard delta G plus RT ln of Q, where Q equals C times D over A times B. So it's the same thing, it's product over reactants. The only difference between Q and K equilibrium is that K equilibrium is that ratio when the system is in equilibrium, and Q is the ratio when the system is at any other point. So Q can equal KEQ, but for most of the time it's not going to. So if we take the natural log of that, multiply it by the gas constant times the ambient temperature, and we add that to the standard delta G, we are going to find the actual delta G for the specific reaction that that we're looking at. And it's just important to keep in mind that when calculating equilibrium constants, you do not use solids or liquids because they have fixed concentrations. So they're not going to be changing throughout the reaction. Water is not going to be changing. If you have, some sort of solid that's in the water that precipitates out, that's not soluble, it's not going to change concentration. Aqueous solutions will change concentration, and so will gases. So those all have to go in the equilibrium equation. Okay, so we were talking a little bit about thermodynamics, and kinetics obviously are also super important for finding out information about these reactions. So thermodynamics will tell us about overall energy changes and the relative stability of the product, but not about the actual rate of the reaction. So when we have spontaneous, we can look at that delta G equation and we can decide if the reaction is spontaneous. So if it's spontaneous, delta G is negative, it's going to tend towards its equilibrium, but it will not, those formulas do not tell us anything about how fast it reaches equilibrium. So we could have a spontaneous reaction that is occurring over hundreds or even thousands of years, or we could have a spontaneous reaction that can be completed 500 times in 1 second. We have to look at kinetics and reaction rates in order to actually get this information. So for a reaction to move forward, the reactants must receive an activation energy to reach the transition state. The transition state is a high energy unstable form that quickly disappears as the product forms. So if we're looking at this diagram, this is called a reaction coordinate diagram. On the y-axis, we have our energy level, and it's very intuitive. So the higher point you are, the higher energy, the lower point, the lower energy. And then it's the reaction coordinate, which is basically, you could basically just say it's sort of the time of the reaction, or how the reaction proceeds forward, more to be more accurate. So we start with the reactants. If we can get activation energy in there through whatever method, we have enough to bump this up. We'll change the molecule just slightly and super, super quickly. It's very transient. It's going to hit its transition state. It's very unstable. And because it's unstable is why this reaction proceeds forward. So it's going to tend toward the products because the products are much more thermodynamically stable than the reactants. So if we can get it right up to this hill, it's going to roll down that way because it's a much steeper fall. All products, not all products, but all reactions, basically the goal in terms of energetics is to get low energy products. We're always moving from high Gibbs energy to low energy. That's just how the universe works. So the transition state is unstable. It quickly disappears as the product forms, and catalysts work to lower the activation energy rate. So the rate of reaction increases. So basically what affects how quickly the reaction will proceed is how much of this activation energy that we actually need. So if it's a higher hump, we're going to need a lot more activation energy. And it's going to take a longer time for these reactions to proceed forward. If we can somehow lower the activation energy, then it's going to be a lot easier to impart energy into the system, and it's going to be a lot easier to drive the reaction forward. And that is what catalysts do. So we have enzymes in our body. Those are the catalysts that usually work at our physiological pH. And what they do is they just bring this activation energy down. So these reactions can proceed much, much more quickly. So catalysts do not affect the thermodynamics of the reaction. Keep that in mind. So if we add a catalyst to this system, we're going to take this bump, we're going to take this hump a little bit down, a little bit farther down, but where the reactants and the products are on this reaction coordinate diagram are not going to change. The actual change, the difference in energy, products minus, or reactants minus products, is always going to stay the same. It doesn't matter how many catalysts we add to the system. So oxidation and reduction, humans store energy in reduced molecules like fats and carbohydrates. So if you ever hear reduced, if a molecule is highly reduced, it's going to be carrying energy. It's carrying high energy. If something is being oxidized or something is oxidized, it is probably lower energy. So if you're oxidizing something, your body oxidizes things to take energy, to transfer energy from that substance to something more usable. And that's basically all cellular respiration is. So as the molecules proceed through cellular respiration, they're oxidized. The energy given off during oxidation powers the regeneration of ATP. So most basically, oxidation is the loss of electrons and reduction is the gain of electrons. So since energy flow follows electron flow, which is something definitely helpful to keep in mind for the MCAT, cellular respiration oxidizes the nutrients absorbed in digestion and transfers this energy to molecules like NAD+ and FAD, which will then use these electrons to power ATP synthase. So that NAD+ and FAD, we refer to them as high energy electron carriers because they're taking these electrons from the nutrients that we're absorbing, and they are carrying them over to the electron transport chain so that we can use this high energy to spin that ATP synthase wheel and couple ADP and inorganic phosphate. So oxidation may appear as a gain of oxygen atoms. It may appear as a loss of hydrogen atoms or the loss of electrons. So technically all three of these it is in the process of losing electrons. It's just not always as obvious as it may have been in general chemistry. So if you gain an oxygen atom, oxygen, you can sort of think of it that oxygen is very electronegative, and it's taking electrons away from the original molecule that it is being added to. So technically that would be considered a loss of electrons. So when we're adding oxygen atoms, or when we're adding bonds to an already existing oxygen atom, we are oxidizing it and we are lowering its energy state. Same thing with the loss of hydrogen atoms. If we're looking at NAD+, that cofactor, if we have NADH and we get rid of the H, we have NAD+. So it's losing a hydrogen atom, but it's also losing that electron in the process. So that would be considered oxidation as well. It's going from a higher energy state to a lower energy state, or the straight loss of electrons. So if you're looking at a metal, for example, and it's changing an oxidation state and it just loses an electron. So iron, for example, if it goes from Fe2 plus to Fe3 plus, it's losing an electron, it's being oxidized. Reduction may appear as the loss of oxygen atoms, gain of hydrogen atoms, or the gain of electrons. So the exact same thing. Loss of oxygen atom or the reduction, the loss of oxygen atoms or the lessening of the amount of bonds on a specific oxygen atom. So if we go from, say, a carboxyl group to a hydroxyl group, we're removing a lot of bonds from oxygen, and technically we're reducing it because we're adding electrons to the molecule. Gain of hydrogen atoms, we can look at NAD plus again. If we add an H, it's a hydride ion. It's not an H plus ion, so that means it's actually carrying an electron, and that's how we get to NADH, which is that high energy reduced form. And the gain of electrons, same thing. If we're looking at a metal, for example, or if we're looking at any sort of ion, anytime it gains an electron and changes its oxidation state, that would be considered reduction as well. Okay, so this is our last review question. Which of the following constitutes an oxidation reaction? Take a minute and a half, 2 minutes, read these, and we will go over it. Okay, so which of the following constitutes an oxidation reaction? So the formation of a disulfide bridge, the formation of ethylene from ethane, NADH converting to NAD+, and changing the hydroxyl group on a molecule to carbonyl group. So the correct answer is D, 1, 2, 3, and 4. These are all oxidation reactions. We're going from a higher energy state to a lower energy state, and we are losing electrons. So let's take a look. The formation of a disulfide bridge, that's a good one because when we're talking about biochemistry, this is a molecule and this is a sort of interaction that always comes up in the context of redox reactions. So if you have two sulfhydryl groups, which is an SH at the end of it, they have that hydrogen on it, they have to lose those hydrogens in order to form a disulfide bridge. So those hydrogens are going to be lost and they're going to take an electron with them because we have to have an empty orbital in order for those disulfide bridges to form. So they have to share those electrons. So they're losing electrons when they form those disulfide bridges. So that would be considered oxidation. The formation of ethylene from ethane, all we're doing is yanking a hydrogen out and we're forming that double bond. Same thing, we need to take electrons out in order for that pi bond to form. So that would also be considered oxidation because it's losing an electron. NADH converting to NAD+. We are losing that hydride, it's taking an electron with it, and it becomes plus because it lost an electron. So that's oxidation as well. And then we have changing the hydroxyl group on a molecule to a carbonyl group. So hydroxyl is a single bonded OH to a carbon, and then the carbonyl group is a double bonded oxygen on that carbon. So if we're adding a bond, we're getting rid of that H and we're adding a bond, that would be considered oxidizing because remember, we're adding more bonds to the oxygen molecule and we're losing electrons. So 1, 2, 3, and 4, those are all correct. So kudos to you if you got that one.